Hey guys, welcome to Zyphie's channel, and today we're doing part 2.2 of Purine synthesis, and today we're forming ATP and a GTP. Well, today we're going to talk about how inositol can form AMP and GMP. So first, let's move on. We have inositol that we formed from the previous video. If you haven't watched it, please check it out. And here we're forming adenyl succinate. Right, how do we do it? Because as you see from the previous video where we have succinate, and of course we're adding a aspartate, and then here what's different is that we don't use ATP to add the aspartate, but instead we use GTP. And then we form this adenyl succinate. And to form adenylate, which is adenylate monophosphate or ATP, ADP, well, those you just need a kinase to add a phosphate. But first we need to form AMP, a base to add the phosphates. So in order for adenyl succinate to change to adenylate, then we get rid of a fumarate. So as before, what's the enzyme that is used to get rid of fumarate? Of course, a lyase. So it's called adenyl succinate lyase. And that's the enzyme. And for the previous part, we have an enzyme that's a synthetase, because we're synthesizing adenyl succinate. So it's called adenyl succinate synthetase. A synthetase, and then you get rid of a succinate, well, a fumarate, called adenyl succinate lyase, an enzyme. And here's what we get, adenylate. And here to continue on, you need kinase to add phosphate groups. And this can be used in RNA. Later, in the next video, we're going to talk about how ATP can transform into DATP. And that's deoxyribose, which is used in DNA synthesis. Now for the second part, we can also transform inositol into phthylate. Well, how do we do that? You can see inositol at a 6 o'clock. You have a double bond CN, but here you get a NH with a C double bond O. So of course, something needs to be oxidized and something needs to be reduced. Redox. And what's the molecules that's often involved in redox of sugars? Well, our dear old friend NAD+, which is reduced to form NADH. And so inosinate is oxidized into dentilate. And what's the enzyme called if we're involved in NADH? Of course, IMP dehydrogenase. So now let's move on to forming guanylate. What do we do? You can see there's a difference at 6 and 5 o'clock. NH. C. Instead, what do we have? An amino group. Amino group, right? So how do we transform this into this? You give, well, of course, our dear old friend, another dear old friend. Who can you guess? It's glutamine, and you hydrolyze it to form NH3, the byproduct. We're gonna use, what do we use? ATP. But this time, products AMP and PPI. You form guanylate monophosphate. Easy peasy, right? And here, well, you can still guess the enzyme name. You're forming GNP. So it's called GNP synthetase. Voila. Here you get the full picture. Full picture of how you can form AMP and GMP. Another kinase to form GTP. They can all be used in RNA. Also in a later video, we're gonna see that special enzyme to form deoxy ones. And now, most importantly, have regulations. Which professor doesn't like to ask you questions on regulations, huh? Remember, in pyrimidine, you form CTP, which inhibits the previous enzyme, which is called aspartate carbamoyl transferase. Right? That's really simple. But here in regulation of purine synthesis, we have ribose 5-phosphate. I'll start for forming PRPP. And we get phosphoribosylamine. And then we just form the inosinate. And then the process that we just did. And then it will succinate form AMP. Salicylate GMP. Right. We reached the point. So here, these are inhibited. This is the inhibit sign by IMP, AMP, and GMP. Why? Because we don't need that anymore if we have a high concentration of IMP, AMP, and GMP. And then NPRPP, right, can be used in pyrimidine syntheses and histidine. And the other two regulation points is this 2 point, which the product itself regulates that part. Because we don't need amine concentration anymore, right? So yeah, guys, this is the regulation of purine synthesis. And last but not least, we have the synthesis of nucleoside diphosphate, which we sort of mentioned here, but let me tell you the key points. Let me change to another fancy color. Nucleoside monophosphate, we add ATP, nucleoside diphosphate, and you add ADP. And what's the enzyme called? Of course, it's nucleoside monophosphate kinase. Because you're giving a phosphate group. And here's an example of how you can make nucleoside diphosphate. Here, let's see. You have AMP, which is adenylate monophosphate, right? And then you add ATP, and you form 2 ADP. Honestly, this process really costs a lot of energy. Like literally making new adenylate phosphate from scratch, de novo synthesis, it takes around 10 ATPs of energy. So this is not cost efficient. And so the condition of why we do this in our body is it's only when we have an extremely low concentration in our body. It extremely low. Because normally in our body we have about like 15 micromole per decimeter cube in our body. It has to be like the deoxynucleotide extremely low that the cell cannot divide. Then we have to extremely low. To a point where cells cannot divide. Or like replication of DNA. Then that's where we use de novo synthesis. Because it takes so much energy, and if we have enough concentration of ATP or the ATP, then why do we need? It's DAMP. Better. That'll be better. Then why do we need to use that much energy to just make our nucleotides? Well, instead we can just use, in a normal condition, we just use the salvage pathway. And that's similar to what we've seen on the first video. Please check it out. Please review that part of this series of biomolecular science videos. It's really important, and because they all interconnect together. And that's all for today's video. I hope you enjoy it, and if I missed anything, please comment down below, and let's make a community to study hard and do well in school. And let's all become study buddies. And hope to see you in the next video. Please subscribe, and please thumbs up and comment on other topics that you would like me to review. I'm currently doing my revision as biomolecular science. It's close. Well, its exams getting closer, so see ya. Bye.